Now, one of the interesting things you found was that this wasn't inherited. Like,
Yes.
So that,
Right.
I found that absolutely baffling. Like, here's this important event that has occurred in these cancers cells,
Yep.
and the daughter cells, I think it was like 25% or roughly
Yep.
a quarter or so.
Yep.
Why is that, and how do you explain it?
So we'll start at the beginning. So what we found is that if you put the enzyme in, so you don't need to maintain the enzyme's levels
Right.
to get it. All you need is an exposure to increased level of the enzyme for less than one cell cycle.
Right.
And what the catch is, it does all of this during S phase. So S phase is when all the DNA is replicated. Cells that are not replicating or proliferating will not gain. So if we arrest cells and they're not dividing, you don't get the gain.
So is there a sampling artifact to a certain extent in terms of number of cells in S phase?
Yes.
And
Because approximately, depending on the cell type,
right.
will determine the percent of cells in S phase. So we see 17 to 25%, and that, if you do FACS, that's close to what that population would represent. The way we sorted this out was you arrest cells, you take a stable population that expresses the enzyme at a fixed level, you arrest it so it's not dividing, so it's at the entry to S, your replication. You don't see copy. If you release them and allow them to proceed into replication, all of a sudden these gains start to emerge.
So the phenotype of being able to overexpress KDM4A
Yep.
exists in these cells,
Yes.
and that gets inherited.
Yes.
But they're not always.
Generating
expressing.
these fragments. So now the million-dollar question is if you look at the rate of proliferation within a
Yeah.
tumor, does that contribute to the heterogeneity of copy number? So for example, if you have a portion of the tumor is rapidly proliferating, and you
Right.
take that into account based on looking at copy gains of regions,
Right.
by chance, does that actually explain why you may see an increased frequency of gains versus other regions of a tumor?
Right.
Because when people do fish on tumors, they often, or even in panels of cells, they see some heterogeneity.
Yeah.
What we would propose is that this may be a function of the cell cycle, or the regulation of the enzyme. When is the enzyme highest, or when is the enzyme exerting its effect? Not just this enzyme, but others that we're looking at. So this may mean that there's different times. So that, therefore, when you look at it, you may assume there's a clone of cells over here don't have a copy gain. Oh, there's a clone over here that has copy gain. It actually may be that this is proliferating and these are in S phase and these are not. So it
Wow.
opens up how you might view a population, especially within a heterogeneous sample. This also means that, it opens up the possibility that, just because it's not there doesn't mean it can't occur, right? So it has the ability, it's just not maybe executing the effect. So if a cell goes quiescent or silent
Right.
and still has high levels, it still has that possibility. It just needs to be released from that pressure to now exert its effect.
And the variations on this must be endless. I mean, what was also amazing is that here you have KDM4A, and it affects very specific areas
Yep.
of the histone, but it's like looking in the Pacific Ocean. I mean, there must be gajillions of little enzymatic activities that are very specific to very specific areas of the DNA.
Exactly. So the coolest part about this is there's 4 enzymes in this family, and they all target lysine 9, target 36, to slightly different degrees of efficiency, but they all target these substrates, and there's 4 of them in vertebrates and in humans. If you put them all in cells independently, the only one that regulates these regions is this enzyme. The others don't. And more importantly, if you analyze now cytogenetically, taking 4,000 tumors, look for cytogenetic gain, the only one that causes these regions to pop up significantly, these regions we identified, is KDM4A in tumors. If you look at KDM4B, for example, which we showed in the paper, it's flat pretty much.
Right.
There's no region that correlates with its gain. And so,
At least in the region you looked at.
by using cytogenetics,
Right.
from tumor samples. Now, the question becomes now if you subclassify tumor type and you look at more tumor sets,
Right.
you acquire more samples to look at this, do you now find that this tumor type, when it has increased B, has these regions upregulated? That would be really a very interesting question. The next part of this is the question is, what are the gains doing?
Right.
Because in these these regions, in ovarian cancer, the paper published by Myerson and his colleagues, where they actually identified 1p34 and 1q21 to be the most frequently gained regions, these are the same two regions we're talking about, because KDM4A is on 1p34. It just happens to be on the downslope of where they had identified what they thought was an oncogene.
Wow.
So this gene drives the same region, statistically and in vitro and in cells. Okay? So this means this is linked to this. We don't know what's in there. So when they did their experiment, they looked at genes in there, and they found that anti-apoptotic genes like MCL1, so on and so forth, were upregulated. They protect the cells against stress,
Right.
and therefore that would be a selective way to get advantage. We know that if you have increased levels of this enzyme, you get a faster recovery from S-phase drugs. We published this previously. We demonstrate in this paper if you treat them with doxorubicin, for example, they have less apoptosis.
Right.
We show that in a cell line that has wild-type p53 that the p53 is up, but not all the way up to activate downstream signals.
So in some ways, it
revs the cell up.
yes.
Our theory is that the fragments actually provide almost like an internal immune response. They get the system just genotoxically agitated enough
Right.
that it doesn't go all the way to suppress cycle or to activate apoptosis, but brings it to a threshold. So in development, that could be very important. In cancer cell now takes advantage of it,
Yes.
and under that selective pressure uses it. So that's how we're treating it. So it could be that the gene inside those regions is essential,
Right.
or it could just be the fact that you get fragment gains in cells.
So it could just be a naturally occurring phenomenon.
Naturally occurring event.
And
And
we're over here looking for all these mutations and
Right.
whatnot, and meanwhile all the actions over here in
It could be that there's a physiological trigger, which means that it becomes very important to see what kind of signaling cascades come in to trigger the events. Are there any physiological inputs that drive the events?
Yeah.
And then if you do see those, then you now start to become aware of, maybe mutations in other pathways, various kinase pathways, for example, could propagate this. And it's a connection.
So this is one of those great stories that I love where you think it's going to be this way,
Yeah.
and the whole world thinks it's this way, but it's right all over here, and it was
Right.
under your nose the entire time. You just couldn't see it.
Yeah, and the cool part is this is the first documented case of an enzyme regulating copy gain. The idea was copy gain was random selection or major damage events with selection, but it shows that it can actually occur. It occurs quickly. The best part is it goes away, and so now we do anything to a cell to make those events stay? So in duplication events you get them, and copy gains you also can see them in mitotic spreads. So in some of those events you get it. So now what we're actually hunting for, and we have this very large scale screen using cancer genomics with Gaddy, is we've taken about 250 candidates, and we're actually screening against all the tumors that we have to see if we can find a correlative with loss of function for those targets and acquisition of these various regions. Because now what we may actually identify is a really important oncogene in the sense that once you lose them, if these events are naturally occurring or certain stimuli cause them, they allow these to be integrated. So now you have a new set of targets and a new set of diagnostic enzymes or genes to think about. So this is kind of I would say the next big frontier of this is, can we identify pathways or enzymes or groups that kind of help you keep these acquired gains?
Okay.
Because now you can start to potentially explain why certain regions are actually gained and acquired and transmitted to the next generation. So,
last question for you is, where is the biotech industry in developing inhibitors of these enzymes? And are we 5 years away? Are we 3 years away?
Yeah, this is the coolest part of this. So there's a number of pharmaceutical companies, some in Boston, external. They've devoted a great deal of energy to targeting methylation and demethylation.
Right.
This family of enzyme, KDM4 family as a whole, got some popularity because one of the family members was shown to be amplified in squamous cell carcinoma, medulloblastoma.
Right.
They designed inhibitors, but they were getting some cross-specificity. And so then other things became more prevalent, more targetable, more easily addressed. And when I've talked to pharma, a number of them, one of the statements they had was, we need to know it's an oncological target. I think based on this, because we profiled so many tumors, because we demonstrate a high prevalence of amplification, a strong phenotypic readout that's related to the activity, and you can distinguish it between others, I think now, according to various groups that have talked to me more recently, this may have actually enabled them to justify targeting. So this whole family, and other KDMs or KMTs are clearly a main thrust of a lot of groups. I think we need biology and diagnostics. Where do you look? And what are you looking for? And if we could assemble an assay that's quick. So if you think about this, like I said, we can do this in 24 hours. So this is amendable to rapid assay screening. And you can use stable cells because it is reversible. So if it's reversible, then you can actually screen.
Right.
So I think as well, this enzyme as well as others, you have these tools. Now you give pharma the option, or biotech the option, to kind of move after it.
Awesome. Well, in recognition, at the end, we always hand out our MGH Cancer Center hat.
All right, thank you.
Congratulations and
Thanks.
really outstanding work.
Thank you.
Thanks.
Thanks.